The next paper is Interferon-alpha may impair endothelial repair mechanism in patients with lupus. And the first author is Dr. Reynolds from the University of Manchester, United Kingdom. Dr. Reynolds.
Thank you, and I'd like to thank the organising committee for this opportunity to present this work. It's well recognised that patients with lupus have an increased risk of cardiovascular disease and that this is not entirely explained by traditional cardiovascular risk factors. Our group showed many years ago now that compared to healthy age and sex match controls, patients with SLE have impaired endothelial function. And this is important because it's the first step in the development of clinical cardiovascular disease. We've also demonstrated that compared to healthy controls, endothelial progenitor cells are abnormal in patients with lupus, both in terms of the number of colonies that they're able to form and the size of these colonies. It's important to recognise that there are a variety of different cell types involved in endothelial repair mechanisms, which are characterised predominantly by the time at which they appear in culture. So we can see that in early culture, around 5 to 7 days, the predominant cell population are early EPCs, or circulating angiogenic cells, which are a myeloid-derived population of cells. These secrete potent angiogenic factors, which help to repair damaged endothelium. As time in culture progresses, around 7 to 21 days, there's a more mixed cell population here. But after around 3 weeks of culture, you can isolate an almost pure endothelial progenitor cell population. This is a very different cell type, which is able to differentiate directly into mature endothelium. There's some suggestion from the literature already that interferon-alpha may be important in these endothelial repair mechanisms, and there's a negative association between the expression of an interferon-alpha signature and the number of early EPC colonies. Furthermore, interferon-alpha 2b has been shown in mixed EPC-CAC populations to induce apoptosis of this cell type and to skew the cells towards a non-angiogenic phenotype. Furthermore, abnormal EPCs from lupus patients, their function can be restored by blockade of interferon-alpha in vitro. So the hypothesis for this work was that interferon-alpha impairs the function of CACs, leading to failed endothelial repair. And what we wanted to look at was to characterise the CACs from healthy controls and to determine the effect of interferon-alpha 2b, which is the most pluripotent of the interferons, on the function of these CACs from healthy controls, and to compare the function of these with CACs obtained from lupus patients. So we isolated CACs from density separation of whole blood and plated the PBMC layer on human fibronectin in endothelial conditions for 7 days, which resulted in a cell population, as you can see here. Consistent with recommendations in the literature, we characterise these cells as being positive for both lipid uptake and binding of lectin. And you can also see that during the period of culture from 2 days to 8 days, the number of these cells increases, suggesting that we're isolating a pro-angiogenic population. We then went on to try and characterise these cells in a little more detail. And here you can see RT-PCR data of aortic endothelial cells here, peripheral blood mononuclear cells, and circulating angiogenic cells. And what I'd like to draw your attention to is the fact that these cells express large amounts of myeloid markers, particularly CD163 and CD206, which is consistent with an M2 alternatively activated macrophage phenotype. And it's also worth noting that these cells do not really express any levels of von Willebrand factor, suggesting that they really aren't of endothelial origin. We confirmed the expression of these myeloid markers by immunocytochemistry, and also the ability of these cells to uptake labeled beads demonstrates that they have a phagocytic capacity. We looked at the ability of these cells to form tubules and matrigel, and here you can see that endothelial cells alone form beautiful capillary-like networks, but the CACs here, which are fluorescently labelled in green, do not have the capacity to form these kinds of networks alone. When they're co-cultured with endothelial cells, however, the CACs line up along the sites of vessel formation. We wanted to develop a model of vascular repair looking at the whole pathway from survival of the cells in vitro through to their migration towards sites of vascular damage, adhesion to sites of activated endothelium, and also the angiogenic effect, which is perhaps the most important. So here you can see that interferon-alpha impairs the survival of these cells in vitro, and here are some representative images showing LDL positivity in untreated cells with increasing concentrations of interferon-alpha 2b, and that this occurs in a dose-dependent manner. I draw your attention to this point here, which is a concentration of 0.1 nanograms per mil of interferon-alpha 2b, which we then used for subsequent experiments. We looked at the ability of these cells to migrate towards STF1 in modified Boyden chambers, and here you can see that CACs from lupus patients, the migration is significantly impaired to those obtained from healthy controls. The addition of interferon-alpha to healthy CACs, however, although seems to suggest that there's a trend towards impaired migration, this is not statistically significant in our data. We looked at the ability of these cells to adhere to TNF-activated aortic endothelium and counted the number of cells in random fields, and here you can see healthy CACs preferentially adhere to TNF-activated endothelium, suggesting that the model is valid. But when we add interferon-alpha to these cells, there is a very small increase in adhesion, which is certainly not significant. So finally, we went on to develop an angiogenesis model, so we could look at the functional effect of these cells by using Matrigel and human aortic endothelial cells, which were cultured either in standard growth media or in the supernatant from our CACs. And here you can see the typical cobblestone morphology of endothelial cells transforming into this capillary light network. We took images of this network and then using a series of algorithms, we're able to transform this rather complex image into an image of a single pixel thickness. This then allowed us to try and quantify some of the features of this network, particularly in terms of the number of junctions, the number of branches, and here the number of these closed polygonal loops, and this is what I'm going to focus on. So here you can see some representative images of standard growth media and CA supernatant, and it's very easy to see just by eye that the complexity of the network is significantly increased in the supernatant from CACs, and the data I'm presenting here are these numbers of polygons. So if we look at the function of lupus CACs compared to healthy control, you can see that there is a small but not significant reduction in the angiogenic capacity of these cells. So what about interferon-alpha? So here we can see standard growth media alone, and that supernatant from healthy CACs in the absence of any interferon significantly increases the density of this network. The addition of a low dose of interferon-alpha here for 7 days does not significantly have any impact on the angiogenic capacity of these cells, but when we culture them with very high doses of interferon, you can see that the angiogenic capacity is almost completely removed. So in summary, we've shown that CACs are myeloid cells with an M2 macrophage phenotype, which have the ability to augment angiogenesis in vitro. We've shown that interferon-alpha 2b impairs the survival and the angiogenic capacity of these cells and appears to have a partial inhibition in the migratory function of these cells. We've also shown that CAC migration is significantly impaired in lupus patients, and that may be important for vascular repair. And we'd like to propose that restoration of CAC function may be a novel therapeutic target to reduce cardiovascular burden in these patients. I'd like to thank my supervisory team, colleagues in the laboratory, and the funders for this work. Thank you for your attention.
Thanks, Dr. Reynolds. Before going to the questions, I would like to know if Dr. Mock or and other one from the group is here, because we think his work is not presented. No, Dr. Mock? Well, so you will have extra time, I think. And I think that there were at least one question for Dr. Jacobsen, and maybe we can do it. So, questions for Dr. Reynolds? Yes, please.
Hi, Tim Newalt, Mayo Clinic. So I'm curious about whether you tried any lower doses of interferon-alpha, just because, patients may have levels as high as 100 nanograms per ml, but many do not actually. When you start getting into nanograms per ml, like 10 nanograms per ml, I don't think we've ever observed patients running quite that high. It may differ, between tissues and local concentration, but I'm just curious if you've seen anything on the lower end of type 1 interferon dosing.
So that's a good question. So when we looked at the survival of the cells in vitro, you could see effects at much lower concentrations than 0.1 nanograms per ml. What we wanted to do is try and find a dose that we could potentially see effects. If we're looking for a negative effect of interferon-alpha, then we want to try and find a good dose that's going to have some kind of effect. And I completely take your point that these are probably levels that are way superphysiological that we see in our lupus patients, but that in some ways is part of the nature of, as you know, of in vitro work. But we would like to try and look at some of the lower concentrations, certainly. Thank you. I'm sorry? We haven't done this in scleroderma. We have some colleagues, as you probably know, in Manchester who have a significant scleroderma cohort, and it would be very interesting to know how the function of these cells does compare amongst the different autoimmune diseases. But that's not something we've looked at the moment.
I'm just curious to know whether this would have any impact on wound repair, because we know lupus patients don't have problems, I think, with wound repair. So how do you tie this data with that? Or do you think it doesn't have any relevance?
I think that's a very interesting question. I don't think anybody's really looked at the effects of interferon on epithelial cell function, which is obviously going to be much more important in wound repair. We're focusing predominantly on endothelial cells, but that convince may be something that's worth looking at. But it's not something that I'm aware has been done either in our university or elsewhere.